There we go. Welcome, everybody. This is the third of 4 webinars. My name's Jackie Jacob. I'm one of 2 poultry specialists here at the University of Kentucky. The other one is Dr. Tony Pescatori, who's there. Wave, Tony. We were asked by, after doing a webinar series last year, we were asked to do one on breeding chickens, so the genetics and whatnot, and then having a hatchery. So this 4-part webinar is a follow-up to that. So during the presentation, if you have a question, you can either type it in the chat box or you can put it in the Q&A. I won't be able to see it, but Dr. Pescatori will be monitoring those. If the question is a clarification of something that I said, then he will interrupt me to ask for the clarification. Otherwise, we'll hold the questions for the end. So if you're like me and you think of a question and if you don't put it down, you forget, feel free to type it in the chat box, and we can, if we need to, we can hold it to the end for the questions. So let me share my PowerPoint. Come on. You see that all right, Tony?
Yes.
Okay, so as I said, this was part of an ongoing webinar series on breeding purebred chickens. And it was meant for Kentucky, but everyone is welcome. And most of it is applicable to other states. We've already had 2 webinars. The first was the introduction to poultry genetics. The second was the genetics of various chicken traits, the comb, the feathers on the legs, leg color, plumage, all that kind of stuff, just to give you some examples of the various things that are genetically controlled. All webinars are recorded, including this one, and posted on our YouTube channel, and the YouTube channel is there. And at the end, I will put that into the chat box because I don't think you can click on it when we're talking. If you can, that's good to know too. So just as a reminder of some of the things that we talked about the first 2 webinars, DNA is the nucleus of each cell. It has chromosomes which submit genetic material from one generation to the other, and they come in pairs. The gene is the segment of the DNA that's the basic unit of hereditary. So genes will code for feather color, comb type, and whatnot. And again, they come in pairs, one from each parent. They're made up of strings of nucleotides, and occasionally errors can happen in the placement of those nucleotides, and so proteins are modified and you get mutations, so something looks different in the offspring as a result of a mutation that happened in the DNA. As I said, DNA and RNA are made up of the nucleic acids. Alleles are the different versions of a gene on a chromosome, and the locus is where it is located. And loci is the plural form of locus. So there are different alleles of different forms of a gene. And some genes are on the same chromosome. They're said to be linked. So as a result, they're inherited together. The closer the genes are on the chromosome, the less likely they are to cross over to a different chromosome. The farther away, the more likely they are to do that. But if you remember, if you are selecting for one gene, you may be selecting for another one that you don't know about that is on the same chromosome. So that has to be taken into consideration. And then there are interactions between alleles at the one locus. They can be dominant, recessive, co-dominant, incomplete dominance. We went through all those in the previous ones. So you can review that in the recordings if you don't remember or you weren't there. We also get interaction between different loci. They call it an epistatic effect, where one gene can affect the phenotype that results from a different gene. So lots of interactions going on that we have to be aware of. Production and physical traits can be controlled by more than one gene, and selection for one parameter can affect the genetics for another. So that's always important to remember. So this is the third webinar, and we're going to be talking about breeding programs and selecting the breeders. Breeding chicks is just basically defined as, you know, the practice of mating pairs or group of chickens to produce offspring. How you do it can affect the performance and appearance of the chickens in the flock. So the first thing that you need to do if you're going to have a breeding program is define your goals. And we already had the one question, what is the perfect breeder to select for? Well, that depends on what your goals are. Different goals are gonna have different selection criteria that need to be used. So you first and foremost need to think about what is the goal of your breeding program. Is it just simply to multiply the flock, or is it to select and improve the flock? So do you want to improve the productivity of a heritage breed, for example? If so, what breed and what production factor? Is it a meat thing or an egg thing? For example, the Delaware breed was an endangered breed, and the Livestock Conservancy was looking at a way to try and motivate people to grow Delaware, and Delaware used to be a meat animal, so trying to select for strains that were good meat birds was one motivation that some Delaware breeders had to do that. Some keep Rhode Island Reds, which is a dual purpose bird, but they want to select it more for egg production or select it more for meat production. So you need to think about, you know, what's your goal? Is it to improve, and what is it you want to improve? Do you want to improve the strain that you have for temperament? If so, what behaviors are important? Are you looking for chickens that are really calm, that are good around kids, or that are really aggressive and can protect the rest of the flock from predators? Or what is the temperament that you are looking for? The heritability will be different. You'll need to look at how you're going to assess that and all those different things. So as there are some publications online. This one was a paper from the Livestock Conservancy looking at improving a dual purpose breed for meat production. They have a very excellent article choosing what age do you evaluate at. So growth curves are not the same for all the breeds. So the age to harvest is going to vary depending on what type of breed you're having. Are you going to have a more Cornish breed, which is not as much a dual purpose, but has more meat involved in it? You know, the Delaware versus the Rhode Island Red. At what point do you are you looking to harvest? So are you trying to grow it faster to that harvest? Are you trying to get a larger weight? Are you trying to get it slower growing? And they discuss all that in their paper. They also look at some of the body assessment criteria. So they evaluate with hands-on all the different criteria of a bird before selecting it. So skull width, heart girth, back flatness, length and width of the breadth of the bird, the body depth and capacity, the breast and keel size, the weight and color. Color can affect what the carcass looks like after you remove the feathers. You know, you're going to have a lot of pigment there from the pin feathers. And feed efficiency may be a criteria that you want to look at as well. So you're going to have to monitor feed consumption and growth rate. So you're definitely going to need a scale if you're doing meat production. And they talk about that and show pictures of how to do it in that article. So don't have time to go into that one in big detail, but that is there as an option. They also have one on improving dual purpose breeds. And of course, they're looking for heritage breeds. For egg production, they have an excellent document on it and show you how to evaluate the pelvic bone arch, the keel to pelvic bone spread, the moltability. How long does it take them to molt before they come into production? How much of a fat pad do they have in the abdomen, which is a sign of feed efficiency? How big of a body capacity do they have? At what age do they come into first egg? At what age do they peak? The size of the eggs produced, the length of the production cycle, how long do they keep producing before they need to take a break, and of course, feed efficiency. And there is always the genetics and environment interaction. So under what conditions do you want to measure those things? Light, are you going to light stimulate? What kind of feed are you going to give? Are they free range or confined? Do they have purchase or not? So when you select, you want to select for the traits and the conditions under which you want to keep the flock. So all of those things need to be taken into consideration. And this document does a great overview of that with pictures and whatnot. And then if you want to improve in ranging behavior, again, you have to think about what criteria to use for the selection. And there's some interesting studies out there. This research with Lohmann silver strain of laying hen, they tagged each individual bird with transponders, and they monitored the ranging behavior in an aviary system with an adjacent winter garden. And they documented the daily frequency of passages between the barn and the garden. They looked at how long they stayed in the garden for each visit. And while the frequency of going in and out decreased as they stayed out longer, which makes more sense, you don't have to go in and out if you're staying there, those two parameters can be very different and have different correlations to other production factors. So how are you going to evaluate your breeders for free ranging behavior? So they looked at the heritability of the different conditions. So H squared is heritability. And so the frequency of passages between the barn and the garden had a heritability of 0.21 to 0.32, which is not too bad. The average duration of single visits, heritability was a little bit higher. So you would think if you select for that, you might have more of an impact. The higher the heritability, the more you are able to select for that parameter. All of these parameters are multi-gene involved. It's not a single gene that determines how often they go outside. There are multiple genes involved. And so selecting for that particular trait involves selection for many different genes that are involved. But they found that selecting for free-ranging ability was negatively correlated with egg production. So the more time they spent outside, the lower the egg production. The length of stay outside, which was that one with the highest heritability, had the most strongly negative correlation. So if you were selecting for length of stay outside, at the same time, then you are selecting for reduced egg production. So you have to think about where do you want to go in terms of compromise, because if you're going to select for, in this particular strain anyway, with the free-ranging behavior, you end up doing it for decreased egg production. The correlation between the passage frequency and egg production was not as strong, but it didn't have as much frequency, as much of a heritability as either. So the graphs just give you an idea of, if it's perfectly correlated, if it's got a high degree of negative correlation or a low degree of negative correlation when you plot the length of stay with egg production or the passage frequency with egg production. And then what selection pressure are you going to use? The heavier the selection pressure, the greater the improvement you're going to see. So for example, in flock A, if they kept the top 20%, then the increase in body weight compared to flock B, when they're only keeping 10%, the top 10%, then they're going to have a bigger impact on the overall growth rate of the breeding stock. So you have to think of your criteria as well as the selection pressure that you're going to use, which means you need to hatch out a lot of chicks in order to select the top 10 or 20%. And then again, as I said, remember that, lots of different genes involved and lots of them are on the same chromosome. So selecting for one thing may select for another. And, a mutation on one could affect something with the others. So back to defining your goals. A lot of that was the quantitative. Now, if we're looking at the qualitative, so if you want to develop a new breed, for example, if so, what characteristics are you interested in? This is for show birds, trying to get it registered might be a little hard, but this is what they did back in the day when the monks, for example, selected and created the Chanticleers for Canada in order to have a winter hardy bird that they could raise in Quebec. Maybe you want to develop a new variety of an existing breed. So if so, what trait are you interested in? So if you have you want to come up with a new plumage color, or you want to add feathers to the legs, or take the feathers off the legs, or whatever. What's your goal? What are you trying to affect with your selection criteria? And then maybe it's just to develop a good strain of an existing breed or variety so that you can win poultry shows. So you have a standard of perfection that you're trying to meet. And so you're selecting against that standard, and you keep only the best that show those standards to win the prize winning bird for that breed and variety. So with the standard of perfection, you know, they look at symmetry, weight and size can be important. Condition and vigor, comb, you know, comb size, color, all that kind of thing, beak color, skull and face color, eyes, color of the eyes, the wattles, the earlobes, the feathering on the neck, on the back, on the tail, on the wings, on the breast, body and fluff, legs and toes. So all those different features need to, you know, you need to perfect those for the purebred that you are interested in. If you have a crested or bearded variety, then crest shape and color, beard shape and color. For the modern game, Alleles, Alleles, Chameaux, their actual body station, how they carry their body is extremely important. And so that becomes an important selection criteria. Japanese bantams, again, that's a specific body type that they're trying to get. The Hamburg and the Rosecomb bantams, they want to look at comb shape as well as size and color. And the earlobe shape and color is important. And of course, the Buttercup and La Flèche, they do a lot more value in their evaluations for the comb. And the Yokohamas and the Phoenix, they are the ones with the really long tail. So the tail shape and color are important. So again, define your goal, find your selection criteria. You can't do anything until you have done that. And so you have different options of what you can do in your developing a breeding program. So it could be individual selection. That's mostly for those kinds of traits. If you are doing something that's more quantitative, like growth rate, egg production, egg size, foraging ability, then you might not be looking so much as individuals, but rather as a flock. But for the traits, because you want to win in a poultry show, then individual selection is typically the way to go. And it can be used in traits that are expressed in both genders. It's kind of hard to select roosters on the basis of egg production, because they do carry the genes for those, but of course they don't express them because they don't lay eggs. Progeny testing is also an option, I guess, especially for roosters, you know, evaluating the performance of their daughters to decide whether or not to keep a rooster. And so this is when you have traits expressed in only one gender, such as the egg production. So progeny testing could be it if you're doing a purebred one and there are different feather patterns for males and females, then you might want to evaluate the feather patterns of the daughters before selecting a rooster, for example. So progeny testing could become important. And SIB testing can also be done. So again, it can be expressed in only one generation. So instead of testing the daughters of a rooster, you can test the sisters of a rooster to see how the family is doing in the performance. So, then you can have pen mating with pedigree hatching. So you can have multiple females allowed to mate with a single male in a small flock. You have separate pens for each male, typically a single male with a number of females. Then you know who the male of the offspring, the parent, the father of the offspring is. In order to determine the mother of the offspring, then you can use trap nesting so that when the hen goes in to lay an egg, it traps her in there. And then when you pick up the egg, you can, you know, mark it based on who who laid it, or, so that you know who the mother is, so you can control the pedigrees of the offspring. When I used to work in a quail unit that was for genetics, and Japanese quail or Coturnix quail, each female lays a distinct pattern of egg, and so we could house a male and several females and still know which egg came from which female. But with chickens, that doesn't happen. So trap nesting is usually used to be able to identify the mother that goes with that particular father. You can do flock matings with 20 to 30 males and 250 to 300 females, but the parentage of the offspring is not really going to be known. If you use, 20 to 30 close males and close females, it's more for, selecting family traits for productivity, not so much of an individual selection as a flock selection. Stud mating is sometimes done. The males housed in a pen or coop, and the females are rotated through them. You have to leave the female with the male for, at least 3 weeks before you start collecting eggs. So that's not as efficient in terms of using the eggs produced, because you can't guarantee that the new rooster is the one that fathered, because the sperm can remain viable in the sperm sacs of the female for several weeks after mating. But if you got her there for 3 weeks, then it's first last in, first out. So the male that's been most active with her in those 3 weeks is going to be most likely the father of the offspring. Shift mating, females are housed in one pen and the males are shifted through. Again, you need to discard eggs for the first 2 or 3 weeks after adding that new rooster. A lot of the selection of the method of mating tends to depend on resources available. How many pens do you have that you can use to keep flocks separated? And there are some benefits to inbreeding. You do it carefully, of course, and that's to decrease the heterozygosity. That means so that you start to make it more uniform, so that the genes from both parents are very similar, so that you're not having, one for blue eyes and one for red eyes. That they're more the same. So the advantage is that it concentrates the genes of a superior ancestor, and it can fix a desired type relatively quickly. If you're having something that's showing up as a defect in when you try to show your birds, may result in chickens that are more likely to transmit their traits regularly when they're used for breeding. And it's easier to predict which crosses will work well with an inbred chicken when it's used for breeding, because you have a better understanding of the genotype of the birds. The problem is that you increase the risk of inheriting undesirable characteristics. So a chicken may be heterozygous and the other gene may be a recessive gene that's, has a flaw that doesn't fit with the standard of perfection. And so if you start concentrating those genes, then that one is more likely to rear its head and cause a problem. It raises the change of a chicken inheriting raise the chance, sorry, not change, the chance of a recessive gene with an undesirable trait. And intense inbreeding, which you don't want to do, may result in loss of vigor in the resulting flock. But if done carefully, you can maintain some inbreeding and not have as many of the disadvantages associated with it. Close inbreeding is when you start to get most of the problems. So mating of parents with progeny, or mating between the sibs, you start getting development of these really inbred lines that can cause some problems. But a lot of times you get your inbreed lines and then you cross out with another line to get some more outbreeding. So the whole purpose of outbreeding is to increase the heterozygosity. So it does increase the vigor of the flock, especially when one or both parents are strongly inbred. It draws from a wider genetic pool. It reduces the possibility of offspring inheriting 2 copies of an undesirable recessive, but the offspring are less likely to transmit their own phenotype onto their offspring because of the more varied genetic backgrounds, making it harder to predict what bloodlines will work well with a cross, because the pedigree is a little bit more muddy. Line breeding can also be done with inbreeding with a highly admired sire or dam to increase the number of individuals with those outstanding characteristics. And this can be done if, if you're doing your purebred chickens and the male has different characteristics than the female. So selecting a male line and a female line, and each generation, the progeny have more and more percentage of the gene from the outstanding parent. So you get your female line, your male line, and then when you want to produce your bird for show, you cross the 2. So it attempts to gain the benefits of inbreeding while reducing some of the risks. You accumulate multiple crosses or 1 or more superior ancestors, typically at the 5th generation and beyond. So you're starting to get there. Selective breeding and weeding out undesirable traits over several generations. They can inherit a wider selection of the ancestors' beneficial genes through their good sons and daughters that you've evaluated. And then strain formation is each person's got their own little flock. So mating of double cousins, again, that's a milder form of inbreeding that you can use. Spiral or clan mating, this is mostly if you want to maintain a flock, not so much of an improvement, but maintaining the flock without getting too much inbreeding in a closed flock. So you maintain 3 to 5 clans, and then you rotate mating the males with members of another clan. So females are replenished with females from those matings, and the females always stay with their clan. So it allows you to maintain genetic diversity within a closed flock so that you're not bringing in any birds from outside, which is of course a biosecurity risk. And according to this, the research, a 3-clan system, if properly used, can sustain a flock for up to 20 generations without too much inbreeding. And a 5-clan system can work forever, but you need heavy record keeping to get it to work properly. So it allows you to maintain a good flock, good genetics, without having to bring in outside genetics. Rolling mating is having a pen for each year or generation, and then you roll to mate. It should be mate young to old. So cocks with pullets, cockerels with hens, to maintain the flock going. So all of that requires record keeping, which requires bird identification. So there are multiple ways that you can identify chickens. Wing bands is one. Unfortunately, they tend to fall off, but you can put wing bands, and you can wing bang them at day old, and if you're lucky, they'll stay on till adulthood. You can also get leg bands. The problem is, of course, that the leg will grow, so you have to be careful that you don't cut off the circulation as they get bigger. And then there are adjustable bands that you can take off and put back on to make it bigger. And then there's neck tags, which one of our researchers here uses here. You just get a little gun, you put it through the skin at the back. Again, they can come off too. You can have numbers on them. You can have barcodes, several different ways of identifying the chickens. Some like to use toe hole punches. I don't like to get into that. I just like numbers. And of course, any breeding program requires breeding pens. And so you can come, if you search online, there are lots of ideas of ways to build breeder pens. It depends on the size of the chickens and how many you're putting together and how many different flocks you need. This is actually the facility for a young man at the time on Martha Stewart's page, her blog. They had a guest blogger there, and he has these types of pens. The ones on the right are for his bantams, and he has quite a big barn at his disposal to use to have all these different pens. You can't have a good breeding program without a lot of separate pens for your different breeding flocks. This one was a more outdoors type. So if you're wanting to make sure that they can handle the environment for whatever you are selecting, these are little mobile pens that you can move around, and you have to be careful to make it predator resistant. That chicken wire there for a lot of areas is not strong enough. A lot of the predators in Kentucky would get through that. You would need more hardware cloth to try and keep them out of that. And then you might need to put an electric fence around the whole area, but you can maintain different breeding flocks. So it's going to limit the number of breeds that you can be working on at any one time by the amount of land that you have for your different breeding flocks. This one was another one, and I like this one because when my dad used to breed, he just used those little arches, and he would have trios that he would put out there. They didn't have a fence to go around in. They just had an empty bottom that they would go down in. This one here, they put it into a fenced in area to let them forage a little bit. So it's sort of a upscale version of what my dad used to do, but he used to take those and bring them inside in the winter in Canada, and he could keep his trios being bred year round. And then of course you get the similar to the pasture pens used in some of the egg and meat production systems. Again, you move it along to fresh pasture, although that doesn't look very pasture-y. Trap nests is basically a nest box that has a door which automatically closes when the hen enters to lay an egg. There are many different designs. You can build your own. If you Google it, you'll come up with some different designs that you can use to build trap nests of your own, or you can purchase trap nests. They come out as well with a variety of them that can be used. These ones you would, of course, have to put bedding material in there. So to have a successful breeding program, you need to develop your breeding program ahead of time based on your breeding goals. And if your breeding goals are production oriented versus purebred oriented, that is going to affect how many types of flocks you have, how much breeding, selective breeding is going on. You need to keep detailed records. The more details, the better. So you want to detail everything. So there is some software. The only one that I could find online was zooeasy.com. It does any species of animal, including poultry. A monthly subscription is required. They used to sell it as a standalone software that you could have on your own computer, but it was too costly to maintain the the updates to it as a sold item. So now they do it online only, but it allows you to keep track of all the birds that you have and that you breed and who bred with who, and you can follow their pedigree along. If you're good on Excel spreadsheets, you can use those as well. You can develop your own based on whatever criteria you're using for selection. When my dad used to work with rabbits, each pen had a breeding card on it to keep track of it. I recommend that as well for your chickens, so that each breeding pen has its own card. It tells you the wing band and description of the mother and father and who their mother and father were, and then the number of eggs that you set in the incubator and the number of chicks that hatch out. This is a very basic one. This one is actually meant for pigeons, but you can develop your own that you could use. And of course, it may not be a pair. It may be a small flock, one rooster with several hens. You could probably, depending on it, do up to 9-10 hens per rooster if you need to. This is some I was just playing around with. It's always good to have a hatch record, and we will be talking more about incubation and hatchery management next week. But once you select the eggs from the birds, and if you've used trap nests so you know who the father and mother is, then you can set their eggs and see how many are hatched, and then band all the chicks that hatch and evaluate on day of hatch for a down color, comb type, shank color, whatever your criteria is, for looking at it when they're day-old chicks. And then whatever your breeding program is for what you're evaluating, evaluate them at another age. So before sexual maturity, you can weight the comb type, size, color, the color of the face, the size and color of wattles, all the different things that would be important if you were doing purebreds, or you could weigh the birds if they're for meat every week to see what their growth curves are like, and their their feed consumption, so that you're keeping track of all that data. The more data you have, you can't go back and get it later. So the more you can have, even if you don't think you need it, might be useful to have, and then decide from there what you want to do. It's also important to hatch large numbers of chicks. You're not going to keep all those chicks. This is why we added the fourth webinar on running a hatchery and selling your excess chicks. So especially if you know, near a city, there's a lot of them that might want pullets to keep, but if it doesn't meet your criteria, don't keep it. So hatch large numbers, but keep just a few. So you need to be able to hatch and raise a significant number. We only keep 1 or 2 for every 10 that you raise. So that, you know, if you need to replenish a chicken flock of 10 to 12 chickens, then you need to raise 100 birds to select the top ideal 10 to 12 chickens that you've selected based on your criteria for your breeding program. And then apply the appropriate breeder selection criteria. So again, based on your breeding goals, what is the selection criteria that you are going to use for your flock? And then if you've evaluated them at different stages that they grow, you can pick the prime of your hatch to keep for the next generation. It's critical that you know what you are selecting for. So have a written standard. If you're raising purebreds, you need the what did I do with it? The standard of perfection. I have one right here. So they come up with a standard of perfection, and it will have all the description of what your particular breed or variety is supposed to look like in order to do well at a show. If you're a newbie at it, have a poultry judge or a long-term breeder help you set up your standard criteria for what you want to select for. Do your research if it's purebred on the genes involved, which genes are going to affect which other traits. For example, the rose comb, I think it was the rose, can't remember. Yeah, rose comb for some breeds adversely affects the fertility of the males. So, you know, do your research on the genes that you're interested in for the traits that you want to select for, so that you know what's happening if you're selecting for more than one trait. Individual selection based on phenotypic value, family selection, based on progeny testing, sibling testing, and you have to have pedigrees, especially for purebreds, so that you know the individual's ancestors, and you're more likely to be able to figure out what the outcome of a cross will be. The tandem method is to select for only one trait at a time, and once you've got success, then you relax your concentration for that and move on to another one, and then when that's perfect, you go on to another one, and so on until you have your perfect bird. Independent culling levels, set up a minimum standard for each level for each trait, and any individual below any one or more of the levels for a particular trait are culled, even if they might be superior in just one trait. If they aren't meeting a minimum standard for all traits, then you don't use it. So you can either work on one method, one trait at a time, or have a minimum standard for all the traits and cull anything that doesn't meet that minimum standard. And others like to use a selection index. So perhaps comb has more value than feathers on the shank. So you come up with a selection index, which has a weighted average on the perceived value and the heritability of a particular trait. And then each trait on each bird is scored till you get a total score, and then you decide whether or not to keep the individual based on that selection index. So those are 3 different ways of identifying the phenotypic value of the birds. And set a percentage of males to keep at each egg. depending on what you're selecting for, some, because you don't need as many males as females. Some recommend that you only keep 60% of the males that hatch. So you right off, start culling out the ones that aren't up to your standard. And then by 8 weeks, when they're starting to show puberty a little bit, take down keep only 40% of those. And by the time they reach sexual maturity, just 12 to 15%. So you have more at the beginning and middle, so that, if there's a mortality or anything that happens, you still have some. So you're going to end up with 12 to 15% at the end. Females, of course, you need more of them, so often save all of them except for any unhealthy ones at hatch. And then by 8 weeks, you keep 80% of them. And then you start determining your traits later, what you're going to keep them for. And then you just basically replacing your flock with the number that you need. So, but don't overlook the environment genotype interaction. So the growing and managing conditions can affect the phenotype, especially if you're selecting for egg production, for example. That can affect, the light, the feeding, the ranging, all those different things. Some of the purebreds, the feather plumage color might fade if they're in the sun. And so there's a lot of different things you have to look at for the environment that can affect the genotype, the phenotype that comes from whatever genotype that you have. So that's what I have. Do we have any questions?
Question on, can you select males and females to produce more females, or is that still potluck?
Can you
select more females for the line to produce more females? The answer to that is, if it could be done, it would have been done a long
time ago. Yeah, it's 50-50 most of the time. And there's, there is no genetic selection for trying to get them to produce more more females than males.
I have a question on, how long does it take to make a new breed?
Depends on what kind of breed you want, what kind of traits you're going to make, but many generations. I think the Chanticleer took like 20, 30 years.
It becomes a breed when it breeds true.
Yes.
I have a question on, my criteria is winter hardy bird, egg producer that can become a fryer. I think the person's asking is, what are some of the traits that makes a bird a winter hardy bird?
Well, I talked a little bit about that last week with the Chanticleer. There are some genes that they have identified that are related to metabolism and fat deposition. So I'm not sure how they evaluated that in terms of live performance, but I would imagine it would be the ones that survived the winter. That didn't get frostbite, that yeah.
We had another question. With no previous rooster in the picture, can I start collecting the eggs right away after placing the rooster and hens together?
At least 48 hours afterwards. Got to give her time to get the egg that she had in her out and then produce another one.
And then if you are lighting the rooster and the hens together, you got to give the rooster 3 weeks head start in order to produce sperm, in order to fertilize the hen.
Yeah, you may end up with unfertilized eggs rather than eggs fertilized by a different rooster that was not there. Assuming he's active, yes.
Well,
Thank
no,
God for-
assuming if you're going to light them together, like in a flock, you have to, when you get them to stimulatory light, you have to wait 3 weeks for the male to start catch up to the female. Do you recommend incubators or broody hens to hatch the eggs?
Well, not all hens will go broody, so you would be dependent on the hen to go broody. Incubators, you can collect the more eggs, because when she's broody, she's not laying eggs. When you take the eggs away, she keeps laying. You're going to get more eggs, and you can hatch them all out together. So if you're doing a breeding program, I would recommend an incubator, and not depending on whether or not a hen goes broody. Some purebreds, the hens never will go broody. So that's always important.
For BBS breeds, is it possible to work on the quality, feathers, color for all 3 varieties at once? Our blacks have leakage. While working on that effect, our blues and splashes. I'm working on that. What effect will have that on blues and splashes? Should we have separate pens for each variety?
Yes, different varieties that you want to work on should be separate. The blue is we covered that last week. It's one of those ones that doesn't quite, it has incomplete dominance. So you will get, if you do blues together, you're gonna, you get all sorts of blacks and splashes and stuff. So blue is a special variety, but if you want to work on a particular variety, I would work on them separately and have different flocks for those, different breeding pens for each variety.
Any other questions?
Next week, we will be talking about incubation and running a hatchery in Kentucky.
Is there an optimum age for breeding to produce the most viable eggs?
Well, eggs are going to be viable, typically throughout, depending on how you store them, which we'll talk about next week. But you want to be able to select. So it depends on your criteria. So if you want to select for ones that can lay longer, then having them go as long as possible, so you have that data for which ones to keep. But as long as you've got the rooster there and she's laying, you should have viable eggs. So
but
chicks,
it's important. But it's like a thing like growth rate, you wouldn't want to use pullet eggs because the chicks will be smaller because the chick is 65% of the egg weight. So when you have a pullet egg, you're gonna have smaller chicks. So it's always a good idea to pick a time where you a point where you select from.
Yeah, a lot of it depends on what you're selecting for. So if egg size is important because of chick size or for selling of eggs or whatever.
An example would be if you want to decrease the time to 50% egg production, you want to select your eggs at the beginning of the flock.
So a lot of it, you got to start with what is your goal? What do what do you want? Always plan ahead of time. Think of what the goal is and then work to get there. Glad you enjoyed it. And everyone should have got the link to the YouTube channel. That's the YouTube channel. It has the videos. This is where that video will go up there as well. It also has the recordings from the ones we did last year on a variety of different topics as well. So I think we answered the questions in the Q&A. So Berkeley, your answer was, the perfect bird depends on what you define as perfect. So your breeding goals are going to help you define what that perfect answer is. And the new breeds, it's getting harder and harder to make new breeds. Depends on what you're looking for. So thank you everybody for joining. It is 8 o'clock, so I will stop the recording.